How Persephone handles QTLs and displays them. There are two main ways to get to QTL information within Persephone. One way is if you know the region and the chromosome that you're interested in. For example, in rice, we'll go to rice annotations for Rhiza sativa. We'll go to chromosome 1, and the database contains pre-populated QTLs associated with chromosomes if they exist. These are QTLs that we've brought from public databases that have been mapped to the physical sequence. So as you scroll through these, you can see a little pop-up box that gives you some information about those QTLs. On this screen, you can also right-click and filter QTLs. You can filter by keywords. So for example, if I type RIZ, I'll get all the rhizome, or I can sort by length and only get the ones that I'm interested in. And as you see, as I make selections here, things are being filtered in the background. So what I'll do is I'll just choose a few of these, and now we have a filtered set of QTLs that we can work with. All QTLs, if you just left click on them, you'll get additional information about those QTLs and descriptions that is part of the database. So if you know the chromosome, if you know the region, if you know the gene, you can get to a region quite quickly and see if there's QTLs that have been mapped into that region. The other way to use QTLs is that if you have no preconceived idea of a genomic location for a given gene or a given QTL, the functionality exists to actually search for studies related to certain traits. So for example, once again, we'll use rice. There's a variety of different studies within the database for rice. There's a trait ontology that you can go through to look for what you're interested in. So for example, we'll do drought sensitivity. As you select in these different columns, it'll automatically filter. So within the system currently, I have one study for drought sensitivity, and it's this one from 2001. You select that, and it tells you all of the different QTLs related to that study. So not only does it contain drought sensitivity, but it contains QTLs for a variety of different things as well. I'll select that study, and I'll show those selected maps. And now what you get is you get the 12 chromosomes from rice visualized, and all the QTLs from this single study on the maps. Once again, you can show and hide QTLs. So I was only interested in drought sensitivity. So I'll select drought susceptibility index, and we'll close. And now I have chromosome 4, and I have chromosome 12. So I'll select those by just hitting the control button in the left mouse. So you can see 4 and 12 is highlighted, and we'll display selected maps here at the bottom. And now you go back to the maps tab, and now you have your chromosome 4 and your chromosome 12 and the QTLs that you have. And now you can go into these QTLs, look at the genes, zoom into each one of these, see the genes around those regions. This one falls within a gene, this one has multiple genes, and you can export these genes. You can do additional research on these genes. You can do synteny analysis on these genes. You can blast the protein sequence against other organisms for these genes. There's a variety of different things you can do after that. But this is how you bring up QTLs within the system. Either find it on the chromosome by looking at specific regions of a genome or chromosome, or looking for studies associated with traits. Both work very well. I hope this helps.